Okay, this is the last video in the census chapter. Um, we'll look at another role of horizontal cells and amacrine cells, and this is this is called lateral inhibition. In order to see distinct boundaries rather than smears, I like to really. This is hard to describe in words. Usually, I write it down on a whiteboard to show you this, but what it means is that generally when you get a sensory uh, stimulus. It's sort of like it's strong in the middle and it's weak and go a little bit out, it's weaker, and a little bit out, it's weaker, and a little bit out, it's weaker. So it's like a smear, right? Well, that's not really fine, acute senses. What you want to need nice fine boundaries between sensation here and no sensation right next door. So nice clear boundaries, and that's what lateral inhibition does. So what happens is if light hits a certain visual area in your retina, the horizontal cells and amacrine cells can tell which cells are being stimulated the most, and they're like right in the center, right? And so what they do is they inhibit the photoreceptors that are on the very edge of stimulation, so they're like turned off. So you have right in the middle you have on, and then right next door you have off. So you have nice clear lines, and that's called lateral inhibition. And this happens actually not just in the vision, but it happens in I know it happens in touch also, and it probably happens in smell, and I think it happens in audition too, with different cells. Okay, then let's go to light and dark adaptation. So dark adaptation is going from a brightly lit area to a dark area. And light adaptation is going from a dark area to a lighted area. And so here's the why. I would give the example of you go into to a matinee, movie matinee, and you get there a little late, and it's a bright sunny day, and you go into the movie theater, and you can't see where your seat is, and you sit in somebody's lap because your rods have been completely bleached. The whole time you're in that sunlight, the rods are bleached. You're not even using them. So when you go into dark, you're really essentially almost blind because there's not enough light for your cones, and your rods are bleached, so they can't be used. So you got to wait about 5 minutes for what's called dark vision to return, and then you can actually see much better. And this is called night vision. Um, and then when you go outside into the bright sun again, you sort of like dazzle a little bit because your rods get all bleached all at once, and then your cones take over, and then you're perfectly okay. This is why in the military, when they're on night maneuvers, they use red lenses in their flashlights to look at maps and stuff because red doesn't bleach rods. If you use white light, you would bleach your rod. Just a tiny shot of white light when you're dark adapted means you got to wait 5 minutes to get your night vision back. So you don't lose your night vision. Because if you're in a battle scene, you lose your night vision for 5 minutes, and you could be dead. So slide 65, vision system. We have a dual vision system. We need receptors that are both ultra sensitive but also provide high visual acuity. And it seems like you can't put those in the same photoreceptor. Rods are highly sensitive because of a high degree of convergence into bipolar and ganglion cells. That is, lots of rods go onto one bipolar cell, which turns it on. So it's really sensitive. Cones provide high visual acuity because of a lack of convergence. All those photoreceptors going into one bipolar cells means you don't have good fine boundaries, nice acute vision. Cones, each cone goes to its own bipolar cell, a dedicated bipolar cell. But it takes a lot to turn on a bipolar cell. That's why why cones are not that sensitive. You have to have a light to make the cone turned off enough to turn on a bipolar, and that is why we need two sets of, uh, of photoreceptors to provide both ultra sensitive detection of light and yet high visual acuity and color. Okay, remember the retinal arrangement. Rods are common on the periphery near the ora serrata. They're really rare in the fovea. Cones are exactly opposite. Cones common in fovea. They're rare near the ora serrata. Okay, slide 66. Color vision, again, we've already gone over this. Short, medium, and long wavelength cones. The perception of different hue depends upon how much each type of cone is stimulated by that wavelength of a photon. Go stereoscopic vision. We talked about this when we talked about a long time ago. We talked about human evolution. We have good depth perception, but you have stereoscopic vision. Good depth perception, but limited area, and people can sneak up on us from behind. As opposed to panoramic vision, we have really poor depth perception, but you can see all the way around behind you. So you have good aspects and trade-offs with those two types of vision. Okay, the projection pathway of vision, the last thing we'll look at, we have the optic chiasma that allows hemidecussation. That is, there isn't complete decussation from left eye only to right brain and right brain to right eye to left brain. Both go to both hemispheres. So you have, and this is called hemidecussation. And the optic tracts go to the lateral geniculate nucleus in the thalamus. So remember, vision goes to the thalamus. And then axons of thalamic neurons from optic radiation go to primary visual cortex in the occipital lobe. But some fibers, remember, go to midbrain and pretector regions for visual reflexes and nuclei that controls eye muscles, the superior collicus. And there's also cells in the retina, this is brand new information, that actually detect how much light comes into your eye and goes to the pineal gland and helps regulate how much melatonin you have. Remember, we talked about that in the endocrine chapter. Okay, the last slide shows a visual representation of the projection path, right eye, left eye only, binocular vision, huge lap because you have stereoscopic vision. And here's this hemidecussation. So right eye, part of it goes contralateral to the left hemisphere, and part of it stays ipsilateral to the right hemisphere. Same thing with the left eye. Part ipsilateral, part contralateral, and here's the axon collateral to the superior colliculus for light for visual reflexes.